This is lecture number 5, Electromagnetic Processes in Biology, focusing on molecules for the Bioelectromagnetics course at DGIST. Welcome. Okay, this week we're going to be doing, as I mentioned, electromagnetic processes in biology, focusing on molecules, in particular the biophysics of ion channels, proton pumps, electron transport chain, oxidation reduction, bioelectrochemistry, and electrophysiology. And then next week we will focus on cells and tissues and maybe cover 2 lectures at that time so that we get caught up. These will complete the principles of bioelectromagnetics, and the second half of the course, as you can see, will be a variety of applications, particularly in medicine. We had asked the question earlier, and I know that we answered this before, but why is the sky blue? I wanted to emphasize that many of you gave physics reasons in terms of scattering of light in the sky, and that the shorter wavelengths, the blue light is scattered more than the red light, and that leads to a blue sky. But in fact, the answer is, those are not wrong answers, but the more complete answer is that our visual system is designed to see in the visible spectrum. It does not see in the ultraviolet. And in fact, the ultraviolet light is also scattered. So if we could see ultraviolet, the sky would be more purple as opposed to blue, if we could attribute colors to it. So in other words, it's a combination of physics and biology that answers this question. So we will do a quick review from the last lecture of basic topics in biology, which include biochemistry, cells and tissues, and basic physiology. We will not go over everything. That was a fairly long lecture, but we'll just talk about the highlights. When it comes to biochemistry, we had several functional groups: hydroxyl group, carbonyl group, carboxyl group, amino group, sulfhydryl in proteins, and phosphate groups found in nucleic acids. And these are important functional groups that you should be aware of. We also talked about the central role of water and hydrogen bonds. This is a water molecule, H2O, with the 2 relatively electropositive hydrogens connected to a central oxygen, which is electronegative. And that partial charge of the electronegativity, or difference in electronegativity, enables the formation of hydrogen bonds, which are a interaction between 2 oxygens and an intervening hydrogen. This is a depiction of water, and you can see that the water, even though it's liquid, is hydrogen bonded, and it has a network that is constantly changing. We talked about polymerization, which is the creation of long molecules in biology, and depolymerization, which is the creation of shorter molecules in biology, typically by the loss or gain of water, respectively. We call it dehydration synthesis in the case of polymerization, and hydrolysis in the case of depolymerization. We talked about phospholipids, and that these are highly amphiphilic molecules with a charged end here and a very hydrophobic section of the molecule below, and depicted as this phospholipid symbol, giving rise to these membrane-like structures that are very important in biology. Now, many electrophysiological events, in other words, bioelectromagnetic processes, will occur at the membrane. So it's very important to understand this structure. In particular, it is critical to understand that this lipid bilayer is also interspersed with proteins, transmembrane proteins that are embedded in the membrane or located adjacent to the membrane. And these integral proteins play a very important role in many bioelectromagnetic phenomena, as we will shortly see. So for example, transport proteins are integral membrane proteins, and they have a nanomotor type function. You can see this is a voltage-dependent potassium channel, which we will discuss in more detail in this lecture. And you can see that it's shifting in its shape in order to either be switched on, where it allows the ion to go through, or off, where it blocks the ion from going through that membrane. We talked also about the flow of information in organisms, in particular prokaryotes, where DNA goes to RNA, goes to protein, and in eukaryotes, where that process of DNA to RNA transcription occurs in the nucleus, and then out in the cytoplasm with translation to proteins. We also talked about the basic anabolism, which is the buildup of molecules, and catabolism, which is the breakdown of molecules. A very important concept in biology is that of energy coupling, where we can take thermodynamically unfavorable processes such as transport, mechanical work, chemical work, connect those processes to the hydrolysis of ATP to drive that unfavorable reaction by combination with a very favorable reaction. We talked about the 4 basic tissue types: the epithelial tissue and the neural tissue in the ectoderm, endoderm, and the connective tissue and the muscle tissue in the mesoderm. Another important concept in biology we discussed was that of homeostasis and rheostasis. Homeostasis is the process of keeping things essentially stable in the internal environment or internal milieu of the cell or organism. An example that we provided was that of glucose, where the glucose level remains relatively constant, and increases in the glucose stimulate the release of insulin, which then reduce the level of glucose. And likewise, extra low levels of glucose will then stimulate the release of glucagon, another hormone that actually releases glucose into the bloodstream from the liver, thus bringing the level back up. What rheostasis is the concept of the body being in 2 states. In other words, like a rheostat, where you switch it between 2 levels. And that is also the case with glucose, where we have a fed state and a fasting state. And in the fed state, glucose is coming from the gut and insulin is relatively high. And in the fasting state, where insulin is relatively low, that glucose is coming from the liver. That switching between 2 states we call rheostasis. So homeostasis is the apparent stability, but behind that stability, we see switches in the body system. That's a very important concept. Another important concept is that of how we interact across the membrane. The plasma membrane, as we described, separates the outside from the inside. So we can transport or exchange information as well as matter. And one example of information transport is this G protein linked receptor. You have some molecule on the outside of the cell, and it binds, releases GTP, and causes some other activity on the inside of the cell. And in this case, it's a ion channel, or it could be an enzyme. We can also have matter transport, of which there are several types of transport of external materials into the cell. One is phagocytosis of very relatively large particles. Another one is pinocytosis, which is the regular uptake of external materials by the cell. And the other one is a specific receptor-mediated endocytosis, where specific molecules are brought into the cell. So that's an overview of the biology concepts that we did last week. And now we will proceed to electromagnetic processes in biology, the topic of this lecture. Any questions so far, you can put into the chat room, or you can speak directly. Any questions, or you can email me or message me by Kakao Talk. So now let's talk about the electromagnetic processes in biology. We'll be talking in 3 sections over the course of the coming lectures. The first one is about molecules. The second one will be about cells and tissues. And the third one will be electromagnetic processes at the level of organisms. So we are going from a more microscopic level and going to a higher level. So first we start with molecules, which is one reason why we reviewed biochemistry with the last lecture. We'll talk about the ions and biophysics of ion channels in relation to biology. We'll talk about bioelectrochemistry and electrophysiology. We'll discuss proton pumps, electron transport, and energy production, oxidation reduction processes. So now let's first focus on ions and biophysics of ion channels. So ions are charged elements, charged atoms. They have typically lost a electron. So here's a sodium that atom that loses the outer electron and becomes a sodium plus. Most of you, of course, are familiar with. So a neutral atom losing electrons becomes a cation, and a neutral atom gaining electrons becomes an anion. And these are ions, and they are very prominent in biology. So this is a list of the atomic and ionic radii. So this is the uh, atom lithium and the much smaller lithium ion. You can see the sodium ion here below the lithium, a little bit larger. And then you see the potassium ion, which is even larger than the potassium, than the sodium ion. So the radius in nanometers of the sodium ion is about 0.095 nanometers, and the radius of the potassium ion is about 0.133 nanometers. So some difference there. And these are successive cations, again, the loss of an electron. When you get to the right side of the periodic table with the higher groups in the non-metals, you tend to get anions, or these become more electronegative. We saw that, for example, in water. Oxygen here is relatively electronegative, and it tends not to lose electrons, but to gain electrons. Most of you who are familiar with chemistry will know this in more detail. We're not going to go into the reasons why particular atoms ionize to cations or anions. Suffice it to say that it's related to the quantum mechanical stability of this outer shell. So in summary, we have positive cations on the left side of the periodic table with metals, and we have negative anions on the right side with nonmetals. So what are some of the key ions in biology? So that was a periodic table of all of the ions. The key ones are, as I mentioned, in relation to the membrane, the extracellular ions, in other words, outside the cell, and the intracellular ions, namely inside the cell. So the extracellular ions include the sodium, as I mentioned, the calcium, very important ion. And notice that calcium has two positive charges, so it is, and it's smaller, so it has a very high, relatively high charge density. And we'll discuss the implications of that. And then you have chloride negative ion. On the inside of the cell, you have mostly potassium. So the outside is mostly sodium and chloride, basically salt, and the inside is mostly potassium positive. And the negative charges that roughly balance it out are various organics, nucleotides like DNA is negatively charged, and the intracellular side is roughly negative. We have major channels. Channels involve passive transport, and then we also have active transport. In particular, very important molecule, the sodium potassium ATPase, or the ATP-driven exchanger, that again, remember that energy coupling we talked about, takes ATP, drives sodium outside the cell and potassium inside the cell. And this is how you establish that difference between the outside and the inside. So in summary, large amount of sodium outside, sodium chloride, and a large amount of potassium and negative organics on the inside. And an important significant player will also be the very high charge density calcium. So an important concept in chemistry, as you know, is the concept of diffusion and a chemical gradient. In thermodynamics, we want to maximize the entropy. That's the second law of thermodynamics, which means that unnatural distributions, or I should say unequal distributions, will tend to equilibrate. And so as you can see here, there is an unequal distribution. Inside this membrane, you have a lot of this ion, and outside you have a very low concentration of the ion. And that will, in order to equilibrate them, cause a chemical driving force. If you have the opposite situation with a high concentration outside and a low concentration inside, your chemical driving force will be in the opposite direction. This is the basic concept of diffusion and a chemical gradient. So here you see that in a depiction, and you can see that as this is moving, you have, this is the density, it's more dense here, you see it spread out, and you see a chemical driving force to spread this distribution out. So this chemical driving force can be described by Fick's law of diffusion, which describes passive transport across this membrane. And it's a very simple equation. The diffusion flux of a concentration A in relation to the bulk motion in the x direction is going to be related to the difference in concentrations, delta C as a function of the volume, and the diffusion coefficient across that region or across that membrane. So the flux is mass per area per time, and if this diffusion coefficient is very low, that flux will be low. If the concentration gradient is very high, or there's a big difference, then that flux will be high. And it's, roughly speaking, a linear relationship. So we had a chemical gradient, but we can also have an electrical gradient. And that's a complicated word for voltage, which more people are familiar with. And a voltage describes a force that will drive the movement of charges. And these ions that we describe are indeed charges. So a potential difference of voltage will create a current across a certain region. If it's in a wire, of course, that current will be in that wire. So more voltage, more current, and that's described by Ohm's law. The voltage is equal to the current times the resistance. So if you double the voltage, you double the current, and this is basically the equations that relate to that. So if we have a difference in ions across a membrane and we apply a voltage, then we will actually drive a current. That current is going to be very similar to the J flux that is driven by a chemical gradient. So if we put those two concepts together of a chemical gradient and an electrical gradient, we get essentially an electrochemical gradient. So we had that situation where we had charges across a membrane. We have a chemical gradient that drives ions out, and electrical driving force that drives them in. In particular, the one that we are referring to is potassium. As we said, that potassium has a high concentration inside the cell, potassium ions. Therefore, there'll be a chemical driving force to the outside. But as this starts to equilibrate and positive charges build on the outside, there'll be an electrical driving force because a negative charge will build on the inside, bringing it back into the cell. So the chemical driving force and the electrical driving force end up being in opposite directions. And we call that an electrochemical equilibrium. And that equilibrium is described by the Nernst equation. The Nernst equation will give the membrane voltage or difference in charge across the membrane as a function of the concentrations and several other constants related to the gas constant, Faraday's constant, the number of electrons, and the temperature. We can derive the Nernst equation from the basic thermodynamic equations of the free energy. And we're not going to go through this derivation, but essentially the point here is the Nernst equation is essentially an electrochemical expression of thermodynamic equilibrium. So now let's go to the second topic, bioelectrochemistry and electrophysiology. We set up the concept of the membrane. We set up the concept of an electrochemical gradient. Let's look at that in a little bit more detail. So this Nernst equation is modified by what's called the Goldman-Hodgkin-Katz Katz equation. So this looks fairly complicated, but it's essentially a combination of not just one ion, which is the Nernst equation, or one charge, or one type of charge. It's a additive sum of all the different charges, because in biological systems, we don't just have one charge. We have sodium, we have calcium, we have potassium, and we have chloride. And so this is essentially a sum of all the positive charges, the sum of all the ratio of the negative charges, and this is a familiar logarithmic term from the Nernst equation. So if we apply that to potassium, sodium, and chloride, we get three terms here. We haven't included one for calcium, which is usually not so significant. So you can see that this is essentially the Nernst equation, but with a sum of the ratio of concentrations of the three major ions that we outlined that are important for biology. So it's a Nernst-like, but has a term for each permeant ion. By permeant ion, we mean ion that can traverse the membrane. And these are some of the summary of what the constant and variables are. So the Goldman-Hodgkin-Katz equation is nothing unusual. It's just an extension into biology of the fundamental Nernst equation, which itself is essentially derived from thermodynamics. So we lead to a concept of the resting potential. So as we mentioned, there is high potassium ions inside the cell and high sodium ions outside the cell. The Nernst equation, it should be noted, is dominated by, not dominated, is defined not just by the concentrations, but also by the permeability ratio. And that permeability ratio, or the permeability, excuse me, is going to determine which of these ions becomes more significant of these three. And it turns out that in the resting state, remember I mentioned about rheostasis, the biological systems tend to go between two states. In the resting state, the permeability is dominated by that of potassium. And what happens is that in the action potential, which shifts the cell from negative state to a positive state, the permeability of the membrane shifts to sodium. So that's very fascinating. Why does the membrane, which is this static sort of lipid bilayer, suddenly shift from potassium permeability to sodium permeability? Well, that's not going to be a function of the lipids. The lipids are passive and static. It's a function of those integral membrane proteins, and specifically protein channels, ion channels, for sodium and for potassium. So turning on and off, switching those proteins is what accomplishes this switch from the resting potential to the action potential. Then when you see the action potential, which is the signal from the nerve cell, it will ultimately return back to the resting potential. And that means that the equation reverts to this dominating term. So all of this was done with what's called a patch clamp, these studies to actually measure that voltage. So you have a ground here, then you'd have a little electrode, or I should say capillary, which can create a seal around the membrane with a little bit of suction. And this is the resistance measured. And as you go up after suction, the resistance is high. So you've created just basically a ability to measure the voltage across this. So this is the recording. It's a little bit more complex because you have the resistance of the patch, you have a leak resistance, you have the capacitance of the pipette, the resistance of the pipette, the resistance of the membrane, and then what we're trying to measure ultimately is the electromotive force or the voltage of the membrane. And then you have the ground or reference electrode out here. So this is a rough diagram of the resistances. So the resistance of the pipette and of the patch and of the membrane are in series, and the resistance of the leak is in parallel with that. So if we can determine the resistance of the pipette, the leak resistance, and the patch resistance, through some other calibrating experiments, we observe the resistance of the membrane. Then, if we drive a current through it, remember by Ohm's law, V equals IR, if we know the current, we know the resistance, we can measure the electromotive force. So in this Hodgkin-Huxley model, the lipid bilayer here is this difference in charges across, and you have different resistances across and batteries, if you will, generated by each of these ions. You have the sodium, the potassium, and the chloride. So in the case of potassium, for example, you have the diffusion, the chemical force out, and the electromagnetic force in. They are opposite each other, so you have a electrochemical gradient. You can see that these resistances have a line through it. And for those of you who are familiar with electric circuits, a resistance with a line through it represents a variable resistance. So we can turn on and off this resistance. And if this resistance is extremely high, then we're not going to have a voltage across it. If this resistance is low, in other words, the permeability is high, then we will have a voltage across it. So the way the cell creates different levels of electric force or electric voltage across its membrane is by turning on and off these resistances across the membrane. So this is the basic Hodgkin-Huxley model of the membrane. So we apply this theory and we can see conductance for potassium in a simulated neuron. And in the action potential, we can see that the conductance for sodium goes up in a spike, and the conductance for potassium, which is the blue line, goes up slightly afterwards. So this is switching quickly between a fast voltage-dependent sodium channel opening and a delayed voltage-dependent potassium channel opening. So that protein that I showed you in the beginning, which was that voltage-dependent potassium channel, that's what's responsible for the blue line here. So let's go back to membrane proteins in a little bit more detail. As I mentioned, you have these integral membrane proteins across the bilayer. We can describe the difference between passive transport and active transport. Active transport involves ATP. It's that coupled process that we discussed. And then passive transport involves transport that is thermodynamically driven by a chemical gradient. For example, diffusion or facilitated diffusion in the case of an actual channel. So all of these ion channels, there are many different types. And this leads to some very interesting bioelectromagnetic phenomena. So in the passive case, we have voltage-gated ion channels, we have ligand-gated ion channels, and we have mechanosensitive, sensitive to mechanical changes, ion channels. So the gradient is already established. This is the passive side. So if there's a change in membrane potential, it opens and the ions go through it down its chemical gradient. In the case of a ligand binding, the ion channel opens. In the case of some mechanical stimuli that bend the cell or push the proteins, again, ions will go through. Carrier molecules basically involve a change in conformation. And then finally, we have transport proteins, again, using ATP that use energy to transport against the electrical gradient. So here's another depiction of the types of ion channels. We have ligand-gated ion channels. We have mechanically gated ion channels, for example, some pressure on the membrane. We have ion channels that are always open. Those are the ones that are in the resting potential that we described, driven by potassium permeability. And we have those voltage-gated ion channels. So here's that example of the voltage-gated or voltage-dependent potassium channel. And you can see that in response to a voltage that changes across the neuron, so during the action potential becomes positive, and that will stimulate this to open. And you can see that the open case is here, and closed conformation, open and closed, that allow this ion to go through. So the overall structure of an ion channel typically is an integral membrane protein, as we described, and there are various dimensions. There's the minimum dimension here, given by 3, the opening dimension 4. So these are the channel domains, typically 4 per channel around the protein. The outer vestibule, which is here, the selectivity filter, which is the narrowest point here, how it determines which ions go through, the diameter of the selectivity filter, phosphorylation sites that can manage or control the activity of this, and of course the cell membrane. The most interesting element here is the selectivity filter, number 3. So this is very interesting. Remember we said that sodium ions are smaller than potassium ions. So if you have a ion channel that is big enough to allow potassium to go through, then why isn't sodium going through? Because sodium is smaller. Well, the main reason is the sodium ions being smaller also have a higher charge density. They tend to have a shell of water around them that is more tightly bound than the shell of water around the potassium ion. So certain characteristics of this selectivity filter, it's big enough for potassium ion, it's big enough for sodium ion, but it's not big enough for sodium and its water molecules. And it doesn't have a configuration that strips the water off the sodium ion. So this selectivity filter would be specific for potassium ions. On the other hand, you have some protein that is specific for sodium ions. It is a smaller filter or a bigger one, depends on the configuration of charges. The bigger one would allow sodium with its waters to transport through, and the smaller one would allow sodium without the waters to go through. So now these ion channels, as we mentioned, have typically 4 domains around them. They can be blocked. For example, this is the calcium ion channel. It can be blocked by calcium channel blockers. Calcium is involved in muscle contraction, and these calcium channel blockers can be used to control blood pressure, for example, because muscle contracts in small blood vessels to increase blood pressure. If we can minimize that contraction, that will relax the blood cells and decrease the blood pressure. So calcium channel blockers are an important category of blood pressure medicine, and they act on blocking these calcium channels from acting. So the third concept that I want to talk about, the electromagnetic processes at the level of molecules, is that of proton pumps. So there are a whole variety of proton pumps. What a proton pump does is you have a membrane and a protein, and protons, which are acid, are transported from one side of the membrane to the other by a protein called a proton pump. And these are typically 7 transmembrane helix proteins. They are not channels, but they are able to use a molecule called a retinal that's within them to bind to a proton on one side and eventually eject a proton from the other side. So that's a proton pump. The most famous of these proton pumps is what's called a Bacteriorhodopsin, BR, but there are several others. Another proton pump are photoreceptors, such as rhodopsins, that are highly related. So Halobacterium is a form of purple bacteria. It has this retinal molecule in the Bacteriorhodopsin, and light comes in and light-activated proton pump. It will pump protons outside of the bacteria. And then why is that important? Because this light comes in, pumps protons out, then the protons coming in through an ATPase, the F0, F1 ATPase, because this is coming down a concentration gradient, the energy coupling that we talked about, ATP, which is a not favorable process, it's an unfavorable thermodynamic process, but driven by the proton pump, ADP can go to ATP. So this bacteria directly converts light to ATP, and it's a halobacterium. So this Bacteriorhodopsin is a very special molecule. And you can see this here. This is the Bacteriorhodopsin, 7 transmembrane helices. This is a retinal molecule that's activated upon light. When it goes from a trans to cis transformation by light. This is Planck's constant times the frequency. Protons will be pumped from the cytoplasm to the extracellular side. It's essentially shifting the proton out towards the extracellular side, and when it goes back, it takes a proton and shifts it to the extracellular side. So it's kind of like a motor that's driven by light. So this is, you can see the ground state, activated. Ground state is trans, activated is cis. And that small difference will pump the proton out. So this is a very special molecule that I had actually studied back in 1988, 87, and we published in 89, Tertiary Structure of Bacteriorhodopsin, Positions and Orientations of Helices A and B in the Structural Map as determined by neutron diffraction. That was one of the first membrane protein structures that was produced. So I wanted to summarize again this energy coupling. Energy coupling takes ATP or any other energetically from favorable process, ATP to ADP, or high protons outside to inside, and couples that to an energetically unfavorable process such as transport, mechanical, or chemical work. In the case of the Halobacterium, ATP is produced, chemical work, by a proton gradient. So this proton pump is very important in this concept of chemiosmotic coupling. So here's the proton going out, and the protons coming back to produce ATP. Bacteriorhodopsin pumps protons out based on light. This comes into the cell, ATP is produced. These are bacteria, but ultimately these bacteria, it's a long evolutionary discussion, but become the mitochondria in eukaryotic cells that have adapted these proton pumps not to work with light, but rather with high energy electrons derived from food nutrients. So that's the last topic we're going to talk today, which is that of electron transport chain and oxidation reduction processes. So what is oxidation reduction? Most of you who are in chemistry are familiar with this concept, of course. Oxidation is the loss of electrons. We call something that has lost electrons as being oxidized. It's a little bit like ionization, not exactly the same, but if it becomes fully lost of electrons, it becomes more positive, and that becomes then a reducing agent. When we talk about reduction, that substance gains electrons. We say that that has become reduced, becomes more negative, and then it can become an oxidizing agent. So there is a inverse relationship. Those things that are oxidized will be reducing agents. Things that are reduced will be oxidizing agents. So here we have an oxidizing agent becoming reduced, and we have a reducing agent becoming oxidized. The reducing agent gives up the electrons, the oxidizing agent gains electrons. So this process is called reduction. This process is called oxidation. And they occur together. The electrons don't just float in the air. There has to be a transfer between one substance and the other substance. And so oxidation and reduction occurs in pairs. Of course, one reason why oxygen, the word oxygen is so related to this oxidation is that one of the most powerful oxidizing agents in natural conditions, not the most powerful, but one of the most powerful, is in fact oxygen. Oxygen, as you know, is a non-metal on the right side of the periodic table. It tends to gain electrons, and thus an oxidizing agent like oxygen tends to gain electrons and become reduced. So why is this all very important? Well, let's go first to sugar and carbohydrates. We talk about this in the biology section. Carbohydrates are CmH2ON, and we have 5 carbon sugars called pentoses, 6 carbon sugars called hexoses, and these are aldoses, these are ketoses. Aldoses have the aldehyde at one end, ketoses have a ketone in the middle here. This is the ring form of glucose, and glucose, as you know, is a very important nutrient molecule. So why is glucose so important? Well, let's look at oxidation and reduction of carbon compounds. The most oxidized state of carbon is carbon dioxide. If you combine carbon with oxygen, which I said is a strong oxidizing agent, that carbon becomes oxidized. And so carbon has the lowest share of electron pairs. And in this case, the electrons don't go directly from one atom to the other, but are shifted. So the way to imagine this is that carbon, the electrons in carbon dioxide, are shifted outwards. They are closer to the oxygens. This is a very important concept because if this is from electricity and magnetism, we discussed this in the physics section, if charges are close together, like charges, that is an unfavorable situation. Like charges repel each other. If charges are dispersed, that is a more lower energy situation. The forces are lower. Charges concentrated, forces are high, energies are high. Charges dissipated or separated, the energies and forces are lower. So in carbon dioxide, the electrons around carbon are dissipated around, and so that represents a low energy form of carbon. Contrast this with methane, which is no oxygens, but just hydrogens around it. The electrons around the carbon will be most concentrated. The carbon, we say, is in its most reduced state. So the highest energy form of carbon is in the fully saturated, fully hydrogenated in this way, carbon. In other words, methane in the case of a single carbon. So the most oxidized is carbon dioxide, the most reduced is methane. And in between we have methanol, formaldehyde, formic acid in terms of successive oxidation. The arrow goes down to suggest the decrease in energy. Where does glucose fit? Well, carbohydrates, such as formaldehyde, which is the one carbon version of sugar, if you will, carbohydrates, glucose are in the middle. That's why they are such an important molecule, because in terms of energy, they are kind of able to be oxidized to carbon dioxide relatively quickly, but they can also be used to go to more complex molecules in anabolism the other direction. The fats, which are related to methane, have the most energy. Most people know that fat has the most calories in terms of nutrients, but it takes some time and complexity to fully oxidize those all the way to carbon dioxide. Of course, in an engine, mechanical engine, internal combustion engine, using glucose is not so efficient because it's relatively oxidized. Oil or gasoline, which is a highly reduced state of carbon, is a better form of fuel, which you burn it up quickly to carbon dioxide. Another very important concept that we need to know is that of thermodynamics. And in thermodynamics, we have reversible processes and irreversible processes. A reversible process is a process that can be reversed in order to attain the initial state of the system, and these are very small changes in the system. In a sense, the initial state and the final state of the system are at equilibrium in the theoretical sense. A fully reversible process is in equilibrium, and it happens in infinite time. In other words, these are theoretical concepts in thermodynamics. No system that occurs in real time that involves a change is a reversible process because it happens in infinite time. An irreversible process is one that cannot be reversed. Finite changes occur in the system, step by step. There is no equilibrium, and it happens in finite time. It happens in a certain period of time. Of course, fully irreversible is something like an explosion. A reversible process is something like an equilibrium. So in thermodynamics, you know that this is a Carnot engine. You get a certain amount of work out there by expansion, compression, and so forth. And if you do it all at once, you get a certain amount of energy. But if you do it in small steps, you approximate this ideal reversible case. This is the reversible case. Infinite stages, you get the maximum work. But if you do it in 3 stages, you get a certain inefficiency. You don't get the full amount of work out of the system. So why is that so important? In the electrochemical reactions in the body, they occur in very small steps. So when you look at the biochemistry and you see all these reactions ultimately going to the oxidation of carbon to carbon dioxide, you see dozens of steps, glycolysis, Krebs cycle, so forth. Some of you have studied biochemistry. You wonder why is it so many steps? Because there are 2 things. One is if you do it all at once, like an explosion, the body burns. So we are not internal combustion engines. We do things in a controlled fashion. Secondly, it is essentially you're getting as close as possible to a reversible process and therefore extracting as much energy as possible. So these electrochemical processes in biology are highly efficient. Not 100% efficient, but certainly more efficient than the explosive properties that we see processes that we see in internal combustion engines. So you have glycolysis, first and second phase. You have the citric acid cycle, ultimately producing carbon dioxide, so getting to that oxidized stage. And as you get to that oxidation, high energy electrons are generated. These high energy electrons then will go down into a gradient and generate ATP. So here's a little bit of a diagram of that. This is a 3-carbon citric acid. Electron carriers are produced ultimately to 2-carbon, 3-carbon, and you lose the carbon dioxide. Electron carriers are created, and those electron carriers will then go to the electron transport chain, which involves very small steps. The electrons go down their energy gradient. Protons are pumped out, so this is reminiscent of Bacteriorhodopsin. Reason why we studied Bacteriorhodopsin, it takes light and transfers that into electrochemical gradient. And those protons come down. The energy coupling allows for the production of ATP. So that is how biological systems create oxidation reduction in very small steps, in a very controlled way, and in a very efficient way. And it's basically utilizing the core laws of thermodynamics. So those are those high energy electrons that are created, protons pumped out of the mitochondrial membrane here. Then the protons come back into the inner mitochondrial matrix in a favorable sense, and then the unfavorable process of the production of ATP occurs. And this shows you a diagram of that. Here are the protons generated by the electron transport chain at a very high concentration outside. The protons come through, ADP creates ATP. Now the protons are coming back in, but then the oxidation through the citric acid cycle to produce oxidation carbon compounds, to produce carbon dioxide, will then pump those protons back out again. So we do a closing thought here. A zoologist, if we're talking about biology, is delighted by the difference between animals, whereas the physiologist would like all animals to work in fundamentally the same way. This is Alan Hodgkin, as in the Hodgkin-Huxley experiment, for which he received the Nobel Prize. And the reason I like this quote is that that action potential and that resting potential that we discussed, the electrochemical changes that we described, and that Hodgkin and his colleagues described with the equations and the experiments and so forth, those fundamental resting potential, action potential processes are essentially the same in all living organisms that have these electrochemical properties, which are basically almost all. There's always an electrochemical gradient across the membrane, generated by energy, by light, by ATP or whatever it may be. That is a fundamental process that is very similar throughout all living organisms. And the details of that are different in terms of the types of proteins. So in dogs or cats or bacteria, the potassium channels are not the same proteins, but they fundamentally do the same process of allowing potassium to go through the membrane. So these proteins are slightly different. Of course, that's one reason why COVID-19 virus, the SARS coronavirus 2, infects humans. It's those membrane proteins that have a particular structure in the human side, but those are very similar proteins to other animals as well. So we end with that quote. And next week, we will focus on cells and tissues and do the neurophysiology, cardiac physiology, how we see photoreception, and cochlear implants, how we can use electrical properties to hear how that process works. And then we'll follow subsequently with electromagnetic processes at the organismal level, which includes bioluminescence, electroreception, magnetoreception, and bioelectric organs. We already did this question, how can we see? But I'll send to all of you the next question for the subsequent week. I got answers from almost everyone. I really appreciate your hard work. I hope everyone is keeping safe. And let me know if you have any questions here. We can discuss in the chat room, or you can let me know by email, and I'll keep this open for questions.